Hello. So today I'm going to be talking about how you can utilize our software Allo to rescue multimap reads, along with the new telomere to telomere genome assembly, human genome assembly, to better understand the regulation of paralogous gene families. So when I had originally joined my lab, my previous experience had all been in transposable elements. And as computational biologists, a lot of what we think about when it comes to transposable elements are their repetitive nature and thus how they can create multimap reads. So what is a multimap read? It's a read that maps to multiple locations in the genome with equally high alignment scores. So your short read aligners like BWA and Bowtie will not be able to distinguish where they actually belong. And this is a big deal because most bioinformatics pipelines, and this is not just only gene regulatory ones, will discard multimap reads. And this actually turns out to be a big portion of your data. And so what we're looking at here is this is about 500 K562 ChIP-seq data sets. And this is just the percentage of multi-mapped reads. And you can see that's quite a lot of your data. So that's a lot of biological significance and money and time that you're throwing away when you throw away multi-mapped reads. And to transfer this into like how many peaks you can rescue if you actually save these multi-map reads, it does vary depending on the ChIP-seq data set, but it can be quite a lot depending on what transcription factor you're looking at. And so I'm just going to go over our method briefly, even though that's not the main portion of this talk, and just talk about how we rescue these multi-map reads in peak-based data types. And so most previous methods that had looked at multi-map reads, what they did is they took the summation of the uniquely mapped read counts around the region and in different ways use the summation. So what we thought is what other information do we have besides just the read counts in these regions? Well, we're looking at peak-based data types, so peaks do have a distinguishable shape. So what if we were able to use that shape to predict whether a read went to a region? And so what we did here is we created images of these regions with the read counts, and then we trained a convolutional neural network to predict peak versus not peak. And it's not really in the scope of this presentation to talk about the training here, but you can read our bioarchives paper if you're interested, which I have linked at the end. But I do want to convince you that we did a decent job in rescuing these multi-map reads before I go on to paralogous gene families. So what we're looking at here is this is about 40 different data sets in 5 different organisms across different assays. So we have neural networks trained for each of these assays that you can use through our software. But this is using read count only, which is the top portion of the neural network or the method that I've shown. And then this is using Allo with the neural network. And as you can see, it outcompetes just using the counts every single time. So the neural network is helping allocate these reads. And we did also compare it to the most recent method for read allocation, for multi-map read allocation called SmartMap. And we were able to outcompete SmartMap in a majority of the data sets that we tested, regardless of the fraction of reads and peak scores. So basically, it's a metric to tell you how good that data set was. And while we were looking at this, we want to do a large metadata analysis to kind of understand where are these peaks at in the genome. And so originally this graph just had the transposable elements on it. And so we assumed that the uniquely mappable peaks and the allo-only peaks, you would see like a really big portion of the allo-only peaks and transposable elements, but that's not what we saw when we looked at this. So you're kind of equally likely to get peaks with uniquely mapped versus allo-only and transposable elements. And I don't have the figure here, but we actually found out that most of the ones that Allo was really good at helping with were young transposable elements and long transposable elements. So we wanted to see were there other regions of the genome that were actually causing these multi-map reads that weren't transposable elements. And so what we did is we overlapped it instead with centromeres, telomeres, and segmentally duplicated genes, and segmentally duplicated genes in particular because they're less, they should be like more similar than some of the other types of gene duplications. And as you can see, they were extremely enriched in the allo-only peaks along with centromeric regions, suggesting that they were a huge source of multi-map reads. And though I'm not an evolutionary biologist, we thought this was extremely interesting and was a gap in the research because it's likely that if so many of these peaks are multi-mapped and people aren't using these multi-map reads, they're missing regulatory activity in these regions by not dealing with multi-mapped reads. So how can we study these better? So there's kind of 2 big things that I think are going to help us study these the regulatory networks of duplicated genes. The first one is we want to use the telomere to telomere genome assembly. And the reason for that is they actually increase the amount of segmentally duplicated regions by 1.6% of the whole genome. So on millions of base pairs, they were able to add in segmentally duplicated regions, suggesting that this assembly should have more of these duplicated genes. On top of that, they have shown that there was an addition of many new genes, and a lot of them are in duplicated gene families. So it's important to use the T to T if you want to like resolve these sections of the genome. The second thing is we need to rescue the multi-map reads with allo based off of the plot I just showed you before and how enriched they are in these allo-only peaks. And we did also extend allo to work with RNA-seq. We're only using the read count only method, which is like a proven method that you can use that will increase the accuracy of allocation. The only thing we did add is allo, whenever you do run it on this, is splice aware. So if you're using star, it'll take the splice junctions into consideration whenever it gets the counts. And then the third thing here is we want to apply this framework to trying to understand this to all ENCODE data sets. But for this talk, I'm only going to show you 16 cell types because that's all we have so far. So to kind of convince you of how many more peaks and how much more information you're going to get using these two different things, the T to T assembly and allo together, what I'm showing here is this is looking at all regions of the genome regardless of duplication status. And you can see that the allo peaks were a modest increase. Only 3% of all peaks were only found with allo for DNA-seq. And the reason I use DNA-seq is I thought it would be a good way to represent just cis regulatory regions in general and not just like one transcription factor like you would get from ChIP-seq. And then with RNA-seq, you do see a modest increase if you use allo to rescue some of these multi-map reads, 7%. But whenever you look at segmentally duplicated regions specifically, you actually see a ginormous amount of them are coming from allo-only peaks, which is not surprising based off of what we had seen in our previous graph with K562 data sets for ChIP-seq. In addition to that, you actually get a lot more regions too using the T to T assembly. So this light gray is uniquely mapped, but only is non-syntenic in H238. And then this is allo, but is non-syntenic in H238. So you're also getting more hopefully properly assembled regions whenever you're using T to T versus just H238. So the first thing we wanted to look at, and like I said, a lot of this is preliminary data, is we wanted to see, a lot of studies have already shown that paralogs are more likely to be regulated together and have similar expression. And so the first thing we wanted to look at is whether or not these paralogous families had similar correlation and expression. And like I said, this is through 16 cell types only, not all of ENCODE, but we did see that this was true. So the background sets we have here are random genes, just comparing the correlation of random genes, and then genes with the same gene ontology term, because you would expect they might have the same like gene regulatory systems. So these are the Q values compared to paralogous gene families that are non-segmentally duplicated, and it is significant against random, but it isn't significant against the gene ontology. When you look at the families that arose from segmental duplications, it's significant for both. One thing that's interesting, I've never seen any of the studies separate the segmental from non-segmental, but when you do put them together, it is significant. So this does follow what previous studies had shown when they were looking at all paralogous families. One thing I wanted to look at too was for the families that had high or low correlation with each other, was there anything that kind of like brought them together? Was there something special about the ones that seemed to diverge a lot once they have duplicated? And it seemed like there was a lot of gene ontology terms related to stress pathways like the GNK cascade, and there was also a lot of things related to like immune response, which was interesting, but also like not surprising that these genes would have to evolve to have a lot of different like types of stress responses or immune responses. And so I did talk about how we had DNA-seq. And one of the problems with looking at DNA-seq and just looking at motifs is I can't say that for sure transcription factor bound there, but I do want to see if the sequences upstream of these different duplicated gene families are more similar. So that's kind of what I wanted to look at here. So what I was looking at is consensus motifs only up to 1 kb. So these are really close. These should be like in the promoter regions of these genes to see if they were more similar than you would expect from random or things with the same gene ontology term. And so it wasn't significant for the paralogous families that were non-segmentally duplicated, but it was significant for the paralogous families that were. One of the issues too, which a lot of people have touched on, even though they were talking about orthologs, when you're talking about alignment of these upstream regions, it's still just as difficult with paralogs because they evolve over time, and the transcription factor motifs are so small that when you do these global alignments, it has a hard time like telling you whether or not those are still there. So the next thing we did, and we want to expand this to all of the ChIP-seq on ENCODE, but I'm just showing you for GM12878, is we wanted to look at whether or not when we look at gene families, were there associations between transcription factors and gene families that you cannot find if you do not deal with multi-mapped reads. And so this is 130 different transcription factors in GM12878. And as you can see, a majority of the associations are not significant, which you would expect. And you do get a lot of significant ones just using uniquely mapped reads. But there were almost 1,200 associations that we found between transcription factors and gene families that you could not find if you did not utilize allo to rescue multi-mapped reads. And what this is showing over here is these 1,000 allo-only peaks. And you can see like quite a lot of them turned out to have a lot of overlap after you use the multi-mapped reads, where some of them even had 100% overlap after using the multi-mapped reads. And I do want to mention too that there were a good few of these that had literature that actually backed that they were associations that were known. So that's good evidence that these should be real associations. And so the big takeaways from my talk is, number one, we developed a software that rescues multi-map reads in various gene regulatory assays. And number two, which I think is definitely the most important thing here, we're always talking about transposable elements when we talked about multi-mapped reads, but it turns out that duplicated genes are a very significant source of multi-mapped reads. And then the third thing, which I hold near and dear to my heart, is don't throw your data away. Whether you use my software or another one or in a different type of omics, I really hope you consider multi-mapped reads. So I just want to thank my lab, and that's the GitHub for Allo if you want to try it, and that's the bioarchives paper for Allo.
Okay, thanks a lot, Alexis. And we have time actually for one or two questions. Yeah, do you want to come up to the microphone?
There.
Okay, cool. Hi. Yeah, that was really interesting. I was wondering if you have a sense for how much coverage you need in order to apply allo, because I was thinking about this would also be really cool to apply to like metagenomic data where you have a lot of like really similar sequences going in there, right?
Yeah, so all of the data sets I've looked at are ENCODE, which does it has really high coverage, like most of their requirements. I've never looked at it for like what you're talking about, so I'm not really sure, to be honest with you, the exact coverage you would need. But obviously, the more, the better you're going to get for that.
Yeah, that makes sense. Thanks.
Sorry.
No, that's fine.